Welcome to Bioratna. Dear students, I am Chandana Sengupta and our today's topic of discussion is Transcription Unit from the chapter Molecular Basis of Inheritance. Transcription Unit. You need to understand the transcription unit properly so that you can follow the transcription process. What is transcription? Transcription is the copying of genetic information from one of the strands of DNA, not both the strands. One of the strands of DNA, that too a part of it, into mRNA. This mRNA will then carry the information and then accordingly the proteins will be formed. I repeat again. What is transcription? Transcription is the copying of, copying of what? Copying of genetic information. From which? From one of the strand of DNA. One of the strand of DNA into what? Into the Xerox copy, that is into mRNA. Out of this one strand of DNA also, it is just a part of it, part of the strand which is being copied. Fine. Now you can easily understand the difference between replication and transcription. In replication, from one parent DNA, two daughter DNAs are formed. मतलब पूरा का पूरा दोनों strand separate हो जाते हैं and new strand is formed on this template strands. Here, out of the two strands, only one strand takes part, that is called template strand, and it's only one part of it is copied. Another difference between DNA and RNA is formed. It is that in DNA it is A, T, C and G are present. These nucleotides are used, whereas during transcription it is A, U, C, G. That means T is not there. Adenine doesn't pair with thymine, rather it pairs with uracil. We will now see why only one of the strand of DNA it takes part in transcription, whereas the other strand is displaced. It does not take part. Both the strands of DNA are not transcribed at the same time. So this is the two DNA strands. Now suppose this much part is to be transcribed. You know that only a part of one segment will be transcribed. When this much part is opening up, then why not both the strands they are being transcribed? Why not two mRNAs are formed? The answer is very simple. First, if these both the strands they transcribe, then what will happen? Then the part of DNA which is transcribing, it will form the two mRNAs. Both of them will form one mRNA. So both will form two mRNA. These mRNA they will form two different types of proteins, and these different types of proteins will have different sequence of different sequence of amino acids. Means so much complication. So many people jumble up at a place to do the work. So this complicates the machinery and that's why this is not feasible. Second thing is that if both the strands they are transcribing, then the thing will not go this far. Means the protein will not be formed. The process will not proceed up to this step. Why? There are two DNA strands. So these two DNA strands, they are complementary to each other. So these are complementary to each other. So the two mRNA which they form, these two mRNA, they will be complementary to each other. So if they are complementary to each other, then they will form double stranded RNA. बंद हो गई information. दोनों एक दूसरे से both have joined together by complementarity rule and they have formed the double stranded RNA. And the whole process of transcription becomes futile. काम बंद हो जाता है. That's why both the strands, they are not transcribed. Fine. Now one very important thing. When the process of transcription has to take place, that means a part of the DNA is copied as mRNA. तो exactly किस point से ये process start हो, exactly किस point पे ये process खत्म हो, the part which is to be transcribed, one, that is structural gene. इसके अलावा हम पढ़ेंगे promoter and terminator and what is their role in defining the coding strand, template strand, etc. Thus students, as transcription is the copying of a part of DNA molecule, part of one strand of DNA into mRNA, so it need to be defined कि DNA का कौन सा part, which part is to be transcribed, कहाँ से शुरू कहाँ से ख़त्म, कहाँ से शुरू और कहाँ पे ख़त्म. That means we are talking about transcription unit. Thus we are talking about transcription unit, which consists of structural gene, three parts. Structural gene, promoter and terminator. That strand which forms mRNA, it is called template strand. Just now I am not going to name anyone, template or coding. Why? You will come to know. One strand out of these two strands, that strand which takes part in transcription, it is called template strand. And that strand which does not code for mRNA, which does not take part in transcription and which is displaced during transcription, that one which does not code for mRNA, it is called coding strand. It is called coding strand. Do you find it strange? See, if this is the template strand, suppose this is the template strand, it is having the nitrogen bases A, G, C, C, G, C. Fine. This is the template strand. So the coding strand, it will have, coding strand is complementary, it will have T, C, G, G, C, G. Now come to mRNA. What about mRNA? mRNA sequence will be exactly same, mRNA codes will be exactly same as like that strand which has not taken part in transcription, except that this T will be replaced by U. So this is U, C, G and G, C, G. See, this is same, this one and this one, it is same. So we can call it coding strand. Fine. Now, which one acts as template and which one acts as coding? That is the question. Enzyme, whether it is DNA polymerase or it is RNA polymerase, they can act only in 5 prime, 3 prime direction. So that strand which allows them to have this point to start from 5 prime, that will be the template strand. Not clear? Just see the diagram. You will say कि both are having, this is strand 1, this is strand 2. Both are having 5 prime 3 prime end. This one is also having 5 prime 3 prime end. So out of these two, which strand will be template strand? This is defined by the position of promoter and terminator. The position of the promoter, it decides which strand will be template and which strand will be coding. Let us see how. Let us see how the position of promoter defines the coding strand and template strand. This is one strand. This is another strand. Fine. And this part, this gene which is to be transcribed, this is called structural gene. One part of transcription unit. So this is structural gene. This is the promoter. This is promoter. What is promoter? Promoter is that sequence in DNA. These are all actually nucleotide sequences, na? The nitrogen based sequences. So here also some nitrogen based sequences are there. So promoter is also a nitrogen based sequence. It is nitrogen based sequence where the enzyme gets attached. Which enzyme? Which enzyme will form mRNA? RNA polymerase. So where RNA polymerase enzyme attaches itself. Enzyme attaches itself. So naturally this is the point. So the transcription will start from this point towards this direction, attaches itself. Now see, this from this direction to this direction transcription has to proceed. Now the new strand needs its 5' polarity for starting. The RNA polymerase, it can act only in 5', 3' direction. So this strand, this upper strand, it is having its 3' on towards the promoter. So naturally here it will provide the 5' end to the mRNA. So this one will become the template strand. So this is the template strand which is providing the 5' end to the mRNA, and this one is the coding strand. So this is the coding strand. Naturally on this side it will be terminator. So terminator is that nitrogen based sequence in the DNA where the process of transcription stops. Just see, by simply exchanging the position of promoter and terminator, the template and coding will be defined differently. How? Let us see this side. This is one strand, this is another strand. The same thing we will do now. This is 3' end, 5. This is 3' end, this is 5. Simply what we are going to do, this promoter is on this side and terminator is on this side. सिर्फ position change कर दिया उसका. Now what will happen? Promoter side में जो जिस strand में 3' end है, that will be the template strand. So this one is the template strand, this one is template, and this one is the coding strand. has it shifted, changed from this one? See, here it was template, here it is coding. Here it is coding, here it has become template. So simply by shifting the position of, interchanging the position of promoter and terminator, the defining of coding strand and template strand, it changes. Let us summarize. You need to have clear idea of transcription unit so that you can understand the process of transcription. We have to study many interesting topics now, like transcription, translation, DNA fingerprinting, HGP, lac operon, etc. First, understand this transcription unit so that transcription becomes easy, then translation will become automatically a easy one. Fine. So, transcription unit, it has three components. The part which is to be transcribed, it is called structural gene. On its one side is promoter, another side it is terminator. Promoter is that region where the RNA polymerase attaches itself, and terminator is that region where the transcription process stops. आपको सिर्फ थोड़ी सी दिक्कत होगी ये समझने के लिए कि इसमें से किसको टेम्पलेट बनाए, किसको कोडिंग बनाए. We say that promoter is present towards the 5' end of structural gene. I repeat, promoter is present towards 5' end of structural gene. तो आप क्वेश्चन कर सकते हो, ये स्ट्रक्चरल जीन के साइड में तो 5' भी है, 3' भी है. एक्चुअली इट इज विथ रेफरेंस टू द कोडिंग स्ट्रैंड. कोडिंग स्ट्रैंड के 5' एंड में प्रोमोटर होता है. टेम्पलेट किसे चूज करना है? that strand which is having its 3' end towards promoter, so that उसका लॉजिक भी अब याद रख लीजिए, so that it can provide the 5' end to the RNA polymerase for the polymerization of the nucleotides. So this was all about the transcription unit. Hope you have understood it properly. If you are new to my channel, then do subscribe, like and hit the bell icon so that you get all the notifications. Thank you for watching the video.